Good morning, students. Today we are starting a new chapter, that is circulation. What is circulation? Circulation is nothing. It is the flow of body fluid. Flow of body fluid is called circulation. So that body fluid is mainly that of blood. It's not only blood is body fluid, so many other things also there. And the main thing we need to discuss is blood. So, what is blood? Blood is a fluid connective tissue. It is a tissue. Tissue means what? Tissue means is a group of cells that perform some common function. So, blood is also a type of tissue. It's a fluid connective tissue. And what is its specific gravity? Specific gravity of blood is 1.05 to 1.06 and its pH is 7.4. Next is about the functions of blood. First function, it transport respiratory gases that you have learned in the respiratory physiology chapter, that the carbon dioxide and oxygen transport takes place with the help of RBC. And next function is transport of nutrients, and through the blood itself all the nutrients that we are eaten after digestion and assimilation, it is taken up by the blood and passing to all the cells in the body. So transport of nutrients is another function. And excretion, blood itself is carrying all the waste to the kidney, and for all elimination blood is playing the main role. Then thermoregulation. Thermoregulation, whatever heat is produced in our various parts of the body, it need to be dissipated out. For dissipating or releasing that heat towards the outside, the blood itself carrying that excess heat to the external body surface by increasing the blood flow to the surface of the body. As that hotness, that increased heat reaches the surface of the skin, it can easily escape to the outside, and that cause the cooling of the body. In that way, it plays a role in thermoregulation. So it is playing a important role in homeothermy by increasing or decreasing the blood flow to the body surface. In that way, it can maintain the body temperature. Okay. Next function is transport of metabolites. So, transporting out all the metabolites means all the things that is metabolized by our body is also transported by blood. The transport of hormones, all the hormones secreted by endocrine glands is distributed by blood, and it plays important role in defense by deploying the antibodies for towards the right location. It is done by RBC and WBC. A part of the blood itself play an important role in immune system that you are going to study in this chapter. Then it plays important role in acid base balance. So many negative and positive charged constituents are present in blood, and so in that way it plays an important role in the acid base balance. It has a buffering. What is a buffer? Buffer is something that maintain the pH without making the medium so much acidic or so much alkali. So, and next last one, it helps in clotting. If whenever a wound is happened in our body, if through the wound, if the blood is continuously start to flow, it may lead to the death of that person. So, in such cases, we need to clot, stop that bleeding. And in such instances, blood plays the constituents of the blood plays an important role. Next slide. Composition of blood. In an average normal person, in his body 5.5 liters of blood is present, and it's a fluid portion is called plasma, and the cellular components are called formed elements. Fluid portion called plasma and cellular components are called formed elements. Then two terms, hematocrit and packed cell volume. This can be asked for 1 mark question also. What is hematocrit value? It is the volume, amount of RBC present in a given volume of blood. Uh, if a given volume of blood is there, and what about the amount, volume of RBC in that particular quantity, that value is called hematocrit. Then what is hematocrit? The percentage of the total blood volume that is occupied by the red blood cells is called hematocrit value, and that value in men is 45% and that in women is 42%. Then what is packed cell volume? It is the volume of formed elements, relative volume. So total, almost all formed elements together, what its volume in blood? That's the packed cell volume. Packed cell volume is determined by centrifugation. It is determined by centrifugation. Centrifugation using centrifuge instrument. And after centrifuging, we can uh, distinguish between all the constituents, all the formed elements, all the constituents of the that taken volume of blood. How it is distributed? First, erythrocyte becomes separated and settled on the bottom of the centrifuge, as it is having much density compared with the other, and it settles in the bottom, and it may commonly accounts for 45%, and that amount, the amount of RBC, that value is called hematocrit. And above that, what settles? Leukocytes, that is WBC settles. And above to the leukocytes, platelets form a thin layer. In this way, after centrifugation of given blood sample, different constituents of blood settles down. First settles down is in the test tube is the RBC, and above that WBC, and above that on the top will plasma. So we are going to learn about each type. This is about plasma. Plasma accounts for 55% of total blood volume, and its color is pale yellowish color. Its composition, what about its composition? 90 to 92% is water, and only the remaining parts are dissolved materials. That dissolved materials can be of organic or inorganic. Next, what are the organic materials of plasma? Main organic materials are of plasma proteins. There are mainly four types: albumins, globulins, fibrinogen, and lipoproteins. And among these four plasma protein, albumin is the abundant one, and it is synthesized by the liver. Next come globulin. Globulin are of three types: alpha, beta, and gamma globulins. Then fibrinogen is there. Fibrinogen is very important for blood clotting. Then lipoproteins also there. They act as carriers for a number of substances like fats, fat-soluble vitamins, hormones, etc. Then other functions of plasma proteins, what are they? They regulate blood pH and also maintain the proper osmotic pressure. Osmotic pressure in the body cells. Now, then non-protein organic contents of plasma. Which are the non-protein organic content? Non-protein, and it's a non-protein, and it is also organic. That part constitute 0.2 to 0.3%. They are mainly of hormones, vitamins, enzymes, glucose, amino acids, nitrogenous base, etc. Then what about inorganic constituents of plasma? Organic we have covered. Then inorganic constituents. Inorganic constituents mainly of salts of sodium. Salts of sodium is the major part, and that salts of sodium mainly are of sodium chloride, sodium bicarbonate, sodium phosphate. Then other salts are of potassium, calcium and magnesium. These are all in less amount. Then serum. What is serum? That also will ask us one more question. Fluid portion of blood, that is plasma itself. Plasma devoid of any type of its solid parts. Only the fluid part of plasma, that is serum. Fluid portion of blood after removing the clot from a coagulated blood. uh, when in the area of a coagulation, a clot, if that clot is removed, we can see a fluidy substance. That substance is called a serum. It is plasma itself. Plasma's fluid portion itself. Next, formed elements. Formed elements are of mainly three types: erythrocytes, leukocytes or WBC, thrombocytes of or blood platelets. There is a balance in its number in our body. Through maintaining a balance between each one of these formed elements, synthesis and destruction. The rate of synthesis and the rate of destruction. There is a balance. By maintaining that balance, the body, its amount, number is keeping constant. First one, erythrocytes, RBC. RBC, first point you have to understand about the nucleus. Except in the case of mammals, in all vertebrates, RBC is nucleated. Then unnucleated RBC is mainly seen in the case of mammals, including human beings. This is the image of RBC. A biconcave shape. Shape is circular biconcave. Biconcave means this side also concave and the other side also concave shape. The only mammal with nucleated RBC is an exception. This is camel. Camel's RBC is nucleated even if it is a mammal. What about the size? Size is 7 to 8 microns in diameter, and in the center it is more thick. There is 1 micron. But towards the periphery it is more thick. There is 2 micron. Then its plasma membrane, there is one specialty. That plasma membrane bears some specific proteins called antigens. The presence of specific type of antigen on the plasma membrane of RBC determine the type of blood. Is it positive? Is it which group? A group, B group, AB? All those things is determined by the presence or absence of these antigens on the plasma membrane of RBC. Then hemoglobin is an important part. It is the carrier. We have learned in the respiratory physiology chapter a very detail about hemoglobin, its structure, its function, everything. So 1/3 of total weight of RBC accounts for hemoglobin, and it is in the inside the RBC, hemoglobin is held by a spongy protoplasmic part matrix called stroma. Stroma is holding the hemoglobin inside the RBC. Next, erythropoiesis means formation of erythrocytes. Erythrocytes is actually formed from the bone marrow. This is the bone marrow. Bone marrow is a spongy tissue seen inside the bones. From this bone marrow cells, actually the RBC is forming. This is the bone marrow cells from first stage. It's called proerythroblast, and then it becomes erythroblast cell, and that erythroblast cell stage, it is with having the nucleus, and then it is changes to a reticulocyte stage for converting from erythroblast to reticulocyte, it need to lose its nucleus and some organelles. And to start the synthesis of hemoglobin. So synthesis starts from hemoglobin. When its synthesis finish, nucleus is discarded and hemoglobin is included, and the RBC, somewhat RBC, gain that reddish color, and still now it is inside the bone marrow itself. When it is fully converted to RBC, only then it is released from bone marrow to the circulating blood. In the final stage, it is converted fully to RBC. In that stage, the remaining organs also that RBC loses, and it is released from bone marrow to the circulating blood system. That about the erythropoiesis. And in adults, the erythropoiesis formation of RBC mainly from bone marrow, as I told you now. But in the case of embryos, first the RBC synthesis is done by gallbladder, and then after some age, after some growth period, then it is taken over by liver, and later on maturity, that function is shifted to the bone marrow. And there's one count also we need to discuss. In the case of women, the RBC number is 4.5 million per cubic mm of blood. Mm means millimeter of blood. In the case of men, it is 5 to 6 million per cubic mm of blood. Then polycythemia. Polycythemia means it's a condition which number of RBC cell in blood is high. Anemia means the RBC count become lower than the normal value. Then lifespan of one RBC cell is 120 days. After that, it will be destroyed. And destruction is done in spleen. Spleen's location is given here, or near to our stomach. And what's the rate of that destruction? About 1% of total body's RBCs replace everybody. So every day. That 1% means how much? 100 billion cells. But 100 billion RBC cells is destroyed, is replaced every day in our body. And all the formation of RBC from bone marrow, that is initiated, stimulated by the help of a hormone called the erythropoietin. Erythropoietin is the hormone that stimulates the formation of RBC from bone marrow tissue cell. Next is leukocytes. Leukocytes number is less when compared with the number of RBC that we already mentioned during the time of composition of blood and its distribution, how its distribution through centrifugation. So leukocytes number is less when compared to RBC's number. Some number is about 4,500 to 11,000 per cubic millimeter of blood. And in resting condition, its number will be very less, and when we do exercise, its number will increase, leukocytes number. Color appears colorless, and there are two types, granulocytes and agranulocytes. That classification mainly depends upon whether the cytoplasm of that WBC contain any granules or devoid granules. Granules are present, that type of WBC are called granulocytes. If that granules are absent, that type of WBC is called agranulocytes. First, we are going to learn about granulocytes. Neutrophil. What are its peculiarities? This picture is given here. First, you listen to this picture. As a round structure, cell membrane is there. Inside a nucleus, we can see the nucleus is not in round shape. Some lobes are visible. So, let's see the peculiarity. They are with the tiny granules inside the cytoplasm. So, they are called granulocytes. They almost stain light violet color. And what about its diameter? 12 to 50 microns is the diameter that we have to by heart. Then nucleus is multi-lobed. Its number of lobes. Nucleus lobes number minimum 3 lobes are present. 3 to how many is possible? Its number is not limited. If a WBC cell have 3 lobe, very easily we can recognize it as a neutrophil. It can be 3, it can be 4, it can be 5, and its number is not clearly mentioned how much. But minimum 3 lobe is present, it will be a neutrophil. And the neutrophil is the most abundant WBC. It accounts 50 to 70 percentage of the total amount of WBC. The next point, what is the function? It is phagocytic in function. It destroys, phagocytes all the foreign body that enters into our body and destroy it. That's the function of neutrophil. It phagocytizes all foreign cells, toxins and viruses. Next is eosinophil. Eosinophil is this one, and here we can see only 2 lobes. And what all other peculiarity? They are with a large acidophilic. Acidophilic means they are acid acid loving. That can be easily stained by acidic stain. That is acidophilic granules. Numerous acidophilic granules can be visible inside this eosinophil cell, and size range from 10 to 15 micrometers. Two-lobed nucleus is present. Then this nucleus lobe number, its type, what is the shape of the nucleus, all these things you have to understand very very clearly, because depending on its difference, and while we are doing some practicals with the blood cells, you are discriminating each type of WBC. So, very clearly you have to understand about the difference between the nucleus among these various WBCs. Here it is 2-lobe nucleus, and its number is less than 5% of leukocytes. In the total volume of leukocyte, it accounts only for 5%. Its function is a weak phagocyte. It can phagocyte and destroy the antigen-antibody complex. And eosinophilia. Eosinophilia means it's a condition when eosinophil cells number increases more than this 5%. Neutropenia is a condition when its level falls below 5%. Falls below the normal value. They are abnormal disease condition. Next, basophil. Basophil means this one. So, picture is given here. Here, the granules, there is one peculiarity. They are basophilic granules, basic stain loving granules, and it stains deep blue to purple color upon staining. And in basophil, these granules are big, and also its number is very high. So it can mask the nucleus, and we couldn't see, we couldn't clearly observe the shape of the nucleus, even if the nucleus shape is horseshoe shaped here, or some way we can say it as lobed. But on viewing through the normal light microscope, we cannot understand the shape of the nucleus because of this abundance of granules, big granules inside the base of it. So its size is 10 to 15 micron diameter, and its granules contains histamines and heparin, and its amount is less than 1% in the total leukocyte count. And abnormal conditions are when its number is high, it indicates hemolytic anemia or chicken pox. Next, agranulocytes. Agranulocytes means these cells are devoid of any type of granules in the cytoplasm. The feature is clear cytoplasm with a single lobed nucleus. Mostly they have a single lobed nucleus, and it is mainly formed from lymph nodes, thymus, tonsils, spleen. From all these organs, they are mainly synthesized. Then first we are learning about the lymphocyte, agranulocyte. Its picture is given here. It's circular. Its nucleus is circular, spherical. Size is 8 to 60 microns diameter. And its nucleus is large, stains dark purple color, and it fill almost the space inside the cell, leaving a narrow a thin rim of cytoplasm around the nucleus. All the other space is enclosed by this large spherical nucleus. Versus function is mainly concerned with the immunity, immune response. Different types of lymphocytes are present: T lymphocytes and B lymphocytes. T lymphocytes attacks the virus infected cells. Then B lymphocytes produce actually the antibodies in the body. And regarding its abundancy, it is the second most abundant WBC. It accounts 25 to 35% of total amount of WBC in the body. In abnormal condition, if it number increases than the normal value, that person is suspected to having chronic infection. And if it number falls below the normal value, that indicates a immune deficiency condition of maybe AIDS. Then monocytes. Monocytes peculiarity, it is the largest WBC. Its picture is given here. It is the largest WBC. Its size is 16 to 22 microns. Largest WBC monocytes. Most abundant WBC is neutrophil. Okay. The nucleus is kidney or bean-shaped nucleus. Function, it is macrophagous against virus and bacteria. And WBC as a whole is concerned with defense in the body. And it show a property called diapedesis. Diapedesis means it can extrude from blood and move to the nearby area to phagocyte any foreign body. That property is called diapedesis. What is meant by diapedesis? Also sometimes asked for examination as one mark question. It is the ability of that monocyte cells to leave the bloodstream and to become a macrophage. Macrophage means to enter foreign particle and to destroy. That property of the monocyte to leave the bloodstream and to become a macrophage is called diapedesis. Its number is 3 to 9% of total WBC. Monocytosis is a condition in which its number rises above the normal value. That condition usually occurs in the stage in malaria and typhoid, endocarditis. All these disease conditions, its number will rise. Then platelets. Platelets is the smallest formed elements. Its size is 2 to 3 microns in diameter. It is fragile, irregularly shaped, and lack spelling mistake. Lack nucleus. Then its function is blood clotting. Blood clotting is the main function of platelets. And its formation is by the fragmentation of bone marrow cells. And that bone marrow cells that is designed to become platelets are called megakaryocytes. And number is this much. You need 10,000. You need 10, 100,000, 10,000 lakh, 2,50,000 to 500,000 per cubic mm of blood. And thrombocytosis means is a condition platelets number is high, and that may result in excessive clotting. Excessive clots can be formed inside the body. Thrombocytopenia is a condition in which platelets count is very low and results in delayed clotting. Even if recurred situation, clotting will not occur. That condition is called thrombocytopenia. So in this way we finish today's section. Thank you.